Hi everyone, today we are talking about glomerular filtration. As you probably know, blood enters the nephron through the afferent arteriole, then flows into the glomerular capillaries. Inside those capillaries, filtration happens. Water and small molecules are pushed from the blood into the Bowman space. The rest of the blood that doesn't get filtered exits through the efferent arteriole. This whole process of moving water and small solutes from the glomerular capillaries into the Bowman's capsule is what we call filtration. And the glomerular filtration rate is how fast this filtration is happening per minute. Now there are several factors that affect filtration, but the most important ones are the hydrostatic pressure and the oncotic pressure. Let's break these down. Hydrostatic pressure is the pressure that water exerts on the wall of blood vessels. Basically, it's pushing things out. Oncotic pressure, on the other hand, is all about osmosis, specifically the osmotic pull created by plasma proteins in the blood. These proteins attract water and try to hold it in the capillaries. That's oncotic pressure. In the glomerulus, we have hydrostatic pressure in the capillaries and also in the Bowman space. And we have oncotic pressure both in glomerular capillaries and Bowman space. Each of these pressures has an effect. Some help push fluid out into the Bowman's capsule and some pull it back into the blood. We also have other important factors that affect glomerular filtration, like the surface area of the capillaries and the permeability of the filtration membrane. Now let's look at the structure of the glomerular capillaries. First, there is a layer of endothelial cells with small pores. These pores allow fluid and small molecules to pass through. Next, there is the basement membrane, which carries negative charge. This negative charge repels negatively charged substances unless they are very small. Then comes the layer of podocytes, which form narrow slits that further filters the fluid. In the end, only very small molecules make it through all three layers and reach the Bowman space. Let's jump into the first major factor that affects the glomerular filtration rate: the capillary hydrostatic pressure. This is one of the main driving forces behind filtration. In the glomerular capillaries, there is pressure ranges between 45 and 60, which is way higher than the pressure in normal systemic capillaries. Now you might ask, why is it so high? Well, there are two important reasons. In systemic circulation, capillaries drain into venules, and the capillary hydrostatic pressure ranges from 15 to 35. On the other hand, glomerular capillaries are drained by arteriole. Venules have much lower hydrostatic pressure in comparison to arterioles. The efferent arteriolar hydrostatic pressure is about 42. For that reason, if the glomerular capillary had lower pressure than 42, there would be no blood flow. Reason number two, the renal arteries supplying the kidneys are short, narrow, and come straight from the abdominal aorta. Because of their short, direct, and relatively small structure, there is less resistance and higher pressure is maintained, which supports strong filtration throughout the glomerular capillaries. Next up, the oncotic pressure. This refers to the osmotic pull exerted by plasma proteins, especially albumin, that draws water back into the capillaries. Let me break it down. If this is our capillary, blood enters the afferent arteriole and exits through the efferent arteriole. As filtration happens, fluid leaves but the proteins stay. So what happens? The protein concentration increases as blood moving along the capillary, which means oncotic pressure rises. At the afferent arteriolar end, the oncotic pressure might be around 25. But by the time blood reaches the efferent arteriolar end, it can rise to 35 or even more. This increasing oncotic pressure acts against filtration, and it's one of the reasons why glomerular filtration rate isn't just sky high all the time. There is a balance. And now let's talk about the hydrostatic pressure of Bowman's space. When fluid is filtered into the Bowman's capsule, it does not just sit there quietly. It pushes back. This pressure is called Bowman's space hydrostatic pressure, and it works against filtration. It's essentially an opposing force, and under normal conditions it's estimated to be about 10 millimeters mercury. There is also something called the oncotic pressure of Bowman's space. But here's the deal. It's zero. Why? Because proteins do not get filtered through the glomerular membrane. So there is no proteins in Bowman's space to pull water osmotically. That's why oncotic pressure equals zero in healthy kidneys. Now let's talk about the filtration membrane itself. Its permeability is a big deal. It has 3 layers: the endothelial cells, the basement membrane, and the podocytes. Together, they decide what gets filtered and what does not. Here is a fun rule of thumb. Anything smaller than 4 nanometers could freely pass. Anything larger than 8 nanometers will not pass. Molecules between 4 and 8 nanometers, well, that depends. It depends on 2 things. Firstly, charge. If it's negatively charged, it gets repelled by the negatively charged basement membrane. And the second factor is the shape. Bulky or irregular shaped molecules have a harder time squeezing through. Now into surface area. It's not just about how wide the capillaries are. We are talking about total filtration surface area of both kidneys. If one kidney is not working properly, the overall surface area drops, and so does the glomerular filtration rate. And here's a cool fact. Mesangial cells, yes, those cells in the glomerulus, can actually control surface area. They can contract to reduce surface area, or they relax to expand the surface area, adjusting how much filtration happens. Let's now put everything together and see how glomerular filtration actually happens. First, we have the hydrostatic pressure of glomerular capillaries, which is 45 mm mercury, and this one favors filtration. Next, we have the oncotic pressure. As we mentioned earlier, near the afferent arteriolar side, it's about 25, and it's about 35 mm mercury on the efferent arteriolar side. And since it pulls water back into the capillaries, it opposes filtration. Then we have the hydrostatic pressure of Bowman's space, which is about 10, and again that opposes filtration. Now let's do the math for the afferent arteriolar end of the glomerular capillary. Hydrostatic pressure of 45, oncotic pressure of minus 25, Bowman space pressure of minus 10. So the net filtration pressure, 45 minus 25 minus 10, which equals 10 mm mercury. That means filtration happens at the afferent arteriolar end. Now let's look at the efferent arteriolar end. Hydrostatic pressure remains 45, but the oncotic pressure increases to 35, and Bowman space pressure still opposes filtration at 10. So the net filtration pressure here is 45 minus 35 minus 10, which equals 0. That means no filtration happens at the efferent arteriolar side. So to summarize, filtration happens at the afferent arteriolar end only. And that wraps up our introduction to net filtration forces. In the next part, we will explore the abnormal conditions that can affect these pressures, and ultimately the glomerular filtration rate. In this part, we are diving into the factors affecting the glomerular filtration rate. We are focusing on the glomerular capillaries. As you know, the capillaries involve 2 main forces: hydrostatic pressure, which favors filtration, and oncotic pressure, which opposes filtration. Hydrostatic pressure is the force exerted by water on the capillary wall. Oncotic pressure is the pulling force created by plasma proteins, mainly albumin, that try to draw water back into the capillaries. Let's start with the hydrostatic pressure. What could affect the hydrostatic pressure inside the glomerular capillaries? First, we have the mean arterial pressure. When blood pressure rises, it naturally increases the renal blood flow to the nephron. And if blood pressure drops, less blood reaches the nephron. But here is the key. The afferent arteriole can regulate this flow, thanks to something called the autoregulation range. Within a pressure range of 80 to 180 mm mercury, the afferent arteriole adjusts itself to maintain a stable GFR. But if blood pressure exceeds 180, filtration and GFR might initially rise. However, too much pressure can cause capillary damage, leading to protein leakage and eventually leading to a renal injury, and even renal failure if untreated. On the other hand, if blood pressure remains below 80 mm mercury for a long time, GFR will decrease, and chronic low pressure could cause ischemia in the nephrons and tubules, increasing the risk of chronic kidney disease. Another factor is afferent arteriolar constriction. When this vessel constricts, less blood enters the glomerulus, which reduces glomerular hydrostatic pressure and GFR drops. Now let's talk about efferent arteriolar constriction. This one has two phases. In the mild stage, efferent arteriolar constriction increases the glomerular pressure, which in turn raises hydrostatic pressure and thus increases the GFR. But in the severe stage, things flip. Severe efferent arteriolar constriction causes very high resistance, which significantly reduces blood flow into the glomerulus. This leads to low plasma flow, and since the flow becomes very slow, the filtered fluid moves faster than proteins, causing the protein accumulation and a rise in oncotic pressure. So now we have low plasma flow, high oncotic pressure, and high resistance. All of this ultimately reduces net filtration pressure, and therefore GFR drops. So we end up with low filtration and low GFR despite high glomerular pressure. And now let's talk about the oncotic pressure. The main role of oncotic pressure is to retain water inside the capillaries and oppose filtration caused by hydrostatic pressure. In the glomerulus, water is constantly being pushed outward due to the hydrostatic pressure. The outward flow is what we call filtration, and it naturally increases the glomerular filtration rate. But plasma proteins, mainly albumin, exert an opposing force, oncotic pressure. They pull water back, preventing excessive filtration. So what happens if something affects the level of plasma proteins? Let's start with a condition called hypoproteinemia, where the plasma protein level drops, especially albumin. With low protein in the blood, there is less pulling force. So water filters out more easily, resulting in a higher GFR. What can cause hypoproteinemia? First, liver disease. Since the liver is responsible for synthesizing plasma proteins, any liver disorder can reduce protein production, will lead to low oncotic pressure. Second, kidney disease. When the kidneys are damaged, especially the glomerulus, proteins start to leak into the urine. This protein loss decreases the amount of protein in the blood, further lowering the oncotic pressure. Third, malnutrition. In stages of prolonged poor nutrition, the body first uses carbohydrates for energy, then fats, and finally proteins. Over time, protein stores become severely depleted, including plasma proteins, which again lowers oncotic pressure. And fourth, malabsorption syndromes. In this case, the small intestine fails to absorb dietary proteins properly. So even after digestion, proteins are lost in the stool rather than entering the bloodstream, which ultimately reduces plasma proteins. All of these lead to low oncotic pressure, which means less resistance to filtration and therefore a higher GFR. And now let's look at the opposite scenario. Situations that increases oncotic pressure and reduces GFR. One example is dehydration. When someone is dehydrated, there is less water in the blood, so plasma proteins become more concentrated. This increases the oncotic pressure, which holds onto the remaining water more tightly, making it harder for water to be filtered out, and as a result, GFR decreases. Thank you so much for watching, and the next video will be an interesting animation. So don't miss it out, and I will see you there.